Welcome back to Founders Day 2020. So after a day of many interesting talks, let's end the scientific session with a debate, which is ultimately the very purpose of science, to trigger debates and discussions. Today we have a rather widely discussed topic: genome editing, genetic modification, and the future of agriculture in India. The conversations around this topic has been so diverse, spanning our scientific, political, social, and even cultural circles. So which better day to discuss this topic than our founder's birthday, who himself had a strong opinion on this topic. So let's open the forum to the varied perspectives of our expert panelists. We invite Dr. Rakesh Mishra, Director, CCMB, Dr. Pooja Bhatnagar, ICRISAT, Hyderabad, Dr. Amitabh Mohanty, NIPGR, New Delhi, Dr. Imran Siddiqi, Group Leader, CCMB. Dr. Raghunand will be moderating this panel discussion. I now request Dr. Raghunand Thirumalai, Principal Scientist, CCMB, to introduce our panelists and to moderate session. Over to Dr. Raghu.
Good evening, everyone, and I welcome you to this debate/panel discussion on genome editing, genetic modification, and the future of agricultural India. As Gayatri just said, there is no better day to discuss this than the birthday of our founder director, Professor P.M. Bhargava. If he was here today, he would be 92 years old, and I'm sure if he was sitting right in the front row and jumping around and excited about the things that will be discussed today. Although he's not with us, his spirit lives on. And the culture that he's set for us as an institution and as a people carries on in all of us who are part of CCMB. So first of all, let me thank the class of 2016 for inviting me to be part of this wonderful discussion as a moderator, and thank you all for coming. And the purpose of this debate, as the email that was sent to me says, is the idea is to discuss the current opinion on what qualifies as a GMO and the differences between genome editing and genetic modification, as well as their merits and demerits, keeping in mind the Indian scenario. With a show of hands, how many of you just understood what I said? Okay, so clearly there are many people in the audience who have heard of genetic modification, who have heard of transgenic organisms, and who are curious about the subject, but many of us are not aware of the technologies and the nuances of the entire domain. So the purpose of this discussion and the elite panel that we have on the stage is to enlighten us about some of the issues that are going on currently, that are being discussed in the field, and to bring out what the issues are, what the possible solutions are, specifically in the context of India. I remember that when I started my PhD in the mid-90s, there was a lot of excitement about transgenic plants. Specifically, BT cotton was being touted as the next big thing, and Monsanto set up a big, very fancy research center next to my institute, and we all got to see its spanking place. We were all very envious of the people who were going to work there, and they started some wonderful work. Then BT cotton got adopted, and then the whole BT brinjal thing came on, and then things started getting a little murky. The excitement kind of dampened, and a lot of things happened. The field seems to have become polarized over the last 25 years. There are people who strongly believe that genetically modified or genetically edited plants are a must for food security for the future. There are those who believe that perhaps our own traditional breeding methods are the way to go, and there are those who are on the fence. And these are all of us, and these are issues that need to be discussed. And things have gotten contentious, sometimes acrimonious, sometimes really nasty. But the point is that we all should continue to talk about this. There's no point hiding in the closet and hoping that something nice will come out of it. And in 2010, the genetically modified brinjal, there was a moratorium on the release of this, although we adopted BT cotton in 2002. In 2017, GM mustard was cleared technologically for commercial trials, but that was halted. So a lot has been happening, and this is an appropriate time to talk about these things, particularly because now the whole new technology of CRISPR and genome editing has come into the domain, and DBT has just issued new guidelines for this technology, and there's a draft genome editing guideline was released last month to be discussed. So these are some of the things that we are going to be discussing today, and we have a distinguished panel to discuss this. And the way this discussion or debate is going to work is each of our panelists will have about 5 to 7 or a few more minutes to make their opening remarks. I will invite them one by one and introduce them to you. And once everybody has made their remarks, we will open the discussion to all of you. We already have a few questions that were submitted by the students, and then we will also take questions from the audience. We have about an hour and maybe 10 minutes for this, so we'll quickly get started. So our first panelist today is our Honorable Director, Rakesh Mishra, who is also Chief Scientist and Group Leader. His laboratory investigates the role of genome organization and nuclear architecture in epigenetic mechanisms and gene expression during development. And most of you are familiar with this, and their model organisms of choice are the fruit fly and zebrafish. For close to 2 decades, his group's research has focused on performing comparative and functional genomics of non-coding DNA, for which they've developed a lot of tools and technologies, and they study the organization and regulation of, I took a lot of time to prepare these bios, so let me studying the organization and regulation of Hox complexes in the context of understanding evolutionary logic of anterior-posterior body axis formation animals. Before I invite Rakesh, I have something cool to tell you. If you go to Rakesh's homepage, there is a section which says awesome quotes by Rakesh. Please go back after this. And I looked at some of those awesome quotes, and some of them was, a couple of them I liked really a lot. And in one he says, I quote, "You cannot be faster than the speed of light and slower than a stationary rock." And being a great believer in using and developing technology for societal benefit, I'm guessing he was talking about technology when he said this: "It is like a road. If you have a car, you can only go where roads are available. What you need is a flying thing to explore places where roads are not made yet." Excellent. So welcome, Rakesh. We look forward to your remarks in this.
Thank you, Raghu. So those quotes are in different contexts, so don't start interpreting them. Okay, so again, once welcome to all of you for this special session. On Founders Day, we used to have public lecture on this day, but this time, for a variety of reasons, we thought we will have this time a public debate and engage audience with this few panelists who have domain knowledge and have been engaging in this discussions and debates and policy issues for a long time. So you are going to have quality information and views coming up. My aim is to give a very general perspective of what we are talking about, especially for the students. And then you can look in that context. So things, the way we do biology is changing, and changing in very major way, largely because of several technologies, and particularly the genome technology and availability of genomic sequences. Some of you may be knowing now there's effort to sequence all eukaryotes on this planet, and program is already on, and India is going to join the program soon. So we are going to have a huge amount of information about the genome of essentially all the organisms. And that is opening a new bioeconomy, new information, and new way of interpreting and learning biological systems. In a way, this is going to liberate us from the model systems, because now, particularly in the view that we can even modify the genomes, now you don't need really model organism. As long as the life cycle is workable, you can actually change the genome and ask what happens. We don't need those traditional systems which are cheap, large numbers, small size, blah, 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 so that you can do genetics with them. Now you can do genetics with pretty much any organism. Now the problem is this, we can today read this information, that we can sequence the genome and we can read this. But do we understand it? Hardly we understand what is the information in the genome. But at the same time, now we have power to change it or edit it. And you can understand how dangerous it is to edit something, a book, without knowing the language, what it means. So you are only going to create either dangerous or worthless things. But shall we wait till we understand these things to perfection and then only touch? Then we will never do it, because the information is so huge. To do experiments, to do understand them in proper classical way, that's not going to happen. So that's why this debate, that what to do now. We have technology, we have ability to introduce these changes in the genome, but we don't understand it much. So we run the risk of being left behind, or we try something what we understand in little controlled way. So that's why we must debate what to do with that. I will just put few things as just information that we can use technology to exercise safety. There are methods of proteomics, metabolomics, transcriptomics, and we know a bit how to interpret them, what they mean, and so on, and at least can have a guess what are the changes that are taking in the system when you tweak one way or other. So we have such technologies one can use and try and make them little bit safer than just doing totally blind. It is very tempting, the new technologies, because you can, so if we are going to discuss today about plants, there is equally exciting and controversial issues for the healthcare system, but we are going to focus on agriculture today. So, for instance, now it is possible to design crop plants which will require less water, they require less resource, they will enhance farmers' income because resources needed will be less, less fertilizer, less insecticide or no insecticide. All these are advantages. If we don't make use of them, maybe we are doing injustice to our farmers, because they need the technology so that they can do the best within the available resources. And these changes in the crop plants can also, you can think that you can break several barriers. You can go to new areas where they don't grow, you can modify, you bring characters from one plant to other, you can make them faster growing, more yield, and so on. And we can even, there are ways of thinking somehow in the context of traditional knowledge we can do. So to do all these things, what we need is proper policy and guidelines so that these things are not misused, farmers' interests are kept in, and so the consumers' interests are kept in mind, and we are not taken for a ride, we are not exploited by bigger players, so that is very important. And in fact, that is the weakness of our system, one of the weaknesses of our system in policy making. Gayatri, during her talk, she brought out this thing that there is no profession called policy analysis, there is no money for policy research, and so forget out policy making. But being a government, you have to pretend that you are making policies, and they do it. I mean, I have little experience very recently. Several kinds of projects you put, and some projects which are of bigger value then go through ministry. Then minister notices, oh, do we have a roadmap, do we have policy, do we have thing? If you don't have, you make it, then only I will sign. So suddenly meeting is called, experts are assembled, and it was like an amusement actually. Half the guys don't understand. They have come with morning flight, they are filling their TA form and they are taking the afternoon flight, they are busy calling the taxi, and by itself they don't know actually what the topic is. They were there because they're director of an institute. So that is the biggest qualification that becomes. And they're sitting there and finally a draft is prepared. So you can see that what is the, and okay, good thing is there are some guys who are sincere, they will sit after that, they will take late flight, they will work and try to, they feel good that they're contributing something. And then that is the draft that goes again to minister. The minister says, okay, now make policy document, national policy document. Now you are given 1 week to make the national policy document. 1 week you are lucky, otherwise they give -2 days. And within that it has to come. So this kind of, we don't have the culture of really working something, studying very well. And so who has to do this? Raghu mentioned that there is a draft document on the website of DBT, RCGM website, which talks about how we are going to, what will be our policy for genetic modifications. And I have access to all information have come. Very little input from public, hardly anything. There are lots of input from foreign companies or organizations of foreign companies, because they want it to be very liberal, they want to be very, there's nothing wrong in being liberal, but you should keep your interest also in mind, because that is more important. These techniques, in my view, are not so dangerous scientifically, they're more dangerous economically, how to handle them. So we have to be more participatory. I can bet that none of you have downloaded the document and seen what it is, though it may be that it will affect our life later, because we'll be eating or wearing those genetically modified materials. So one thing is that the system is not good, I'm frustrated, and then you can just give up everything, come and become a professional critic and just intellectualize and criticize. Doesn't help. The other alternative is that you see what you can do in Desi ecosystem, try to do your best and hope that you can have few more people who are concerned and try to contribute. In the end, like Gayatri mentioned, if you persist long enough, something will come. So that is one approach which I think is better approach than being vociferous, critique and destroy things and go. Then you will actually be sidelined, because system will go. They're not going to stop anything. So the best is to see how we can contribute to that. Now, it's also interesting because genome editing is not being taken as genetic modifications, because many international bodies, many countries had adopted that genome editing is not GM, so it can be used. Do we consider GM and genome editing is the same? Maybe yes, maybe no. So this is, but generally people are in favor that as long as the changes are small, 1-2 nucleotides or something of this kind, we will call it non-GM, like natural mutations. Whether it is a good thing to adopt or bad thing, we should discuss, because these are the things which will have long-term consequences. And other thing we have to see is that people who are doing science, who understand, like in the institution like CCMB or other such organization, they should actively participate. They should put their opinion to the government policy portals, and also talk to people around. GM doesn't mean poison, but that doesn't mean that we just start eating everything GM without knowing what we are talking about. So spreading proper information so that people realize what this thing is very important, and that's why we need to discuss and debate. I think many such events will be required, and I hope that someday we'll come up with what is popular and scientifically solid and economically relevant policy or way forward that we should go. So with that, I'll stop there, and we will maybe hear more specific and more focused points from the other speakers. So thank you.
Thank you, Rakesh, for kickstarting the debate. I didn't mention that Rakesh is also the head of the RCGM, which is a review committee on genetic modification, and he's going to be at the forefront of deciding what the policy is going to be. So thank you for setting the stage. Let me invite our next expert, Dr. Pooja Bhatnagar Mathur, who is a theme leader in cell molecular biology and genetic engineering at the International Crop Research Institute for Semi-Arid Tropics, which we know as ICRISAT, and she's been there since 2006. Dr. Bhatnagar Mathur has over a decade of expertise in genetic engineering of grain legumes to integrate useful traits, including enhanced diseases and pest resistance, abiotic stress tolerance, and nutritional enhancement. She has a special interest in integrating scientific innovations to design and build new biological functions and systems to generate robust breeding pipelines using existing as well as newer genome editing technologies. She is also involved in biosafety assessment of transgenic products and building capacities of stakeholders in different target countries of Asia and Africa to comply with the internationally and nationally laid out biosafety guidelines. And more recently, she's also played a role in the establishment of the recently inaugurated Rapid Gen facility, which I hope she will tell us about a little bit, at ICRISAT, which is designed and incorporates cutting-edge technologies to accelerate crop breeding times by up to 40%. So, Dr. Bhatnagar Mathur, we are delighted that you accepted our invitation to be part of this panel, and we look forward to your thoughts on the subject. Can we have a round of applause for Dr. Bhatnagar Mathur?
A very good afternoon. Thank you so much for a very kind introduction, Raghunan. I am representing ICRISAT here, as he mentioned, and I thought I should go and quickly put some slides together because the audience here probably is not very specific to agriculture, and so I just wanted to communicate what is going on in countries. The topic itself was a little confusing because we are talking of genetic engineering, gene editing, all in one sentence. So I thought, I'll just try to quickly take you through what we do at ICRISAT. So ICRISAT being a CGIAR research organization, we have an international responsibility on bringing all possible tools and technologies in the breeder's toolkit. So considering that, we use these modern tools and technologies for agriculture, for the traits that are difficult to breed for by the crop breeders, and so you will see I have put the word intractable traits, considering that there are a lot of traits in agriculture that are not easy to just breed using conventional agriculture, so we need modern tools and technologies to really address some of those things. A very quick look on where we are, semi-arid tropics. So ICRISAT stands for ICRISAT International Center for Research on Semi-Arid Tropics, and which is, interestingly, the home to most emerging economies, covering almost 6.5 million square kilometers across 55 countries, and with over 2 billion people in these SAT regions, of which 644 million are the poorest of poor. So obviously we are talking about a region, we are talking about the geography, which is the home to the most poorest of poor, including our country. It is there in orange, and there are high levels of poverty, malnutrition, and environmental degradation. So considering we aim in India also, we aim to become a 5 trillion economy in next few years, we still don't have to forget that we have the responsibility for people who are residing in these SAT regions, and they would need innovations in agriculture to really, have their the livelihoods. So the strategic questions then would obviously be, what are the constraints to the productivity of those systems that we are talking about, and what existing or potential technologies might best address those constraints, and under what scenarios. A very quick quick snapshot of from breeding to accelerated breeding. So agriculture is not new. It has been happening for centuries now. The conventional breeding that we now call it as is actually the way agriculture has been done since ages, from the first domestication for plant when it actually began. But then the fact is there has been, the agriculture has been narrowing the genetic diversity. The mutation breeding again brings another dimension to it, increases the genetic variability that we otherwise see getting reduced day by day with conventional breeding. However, this would involve methods like either chemical mutagenesis, which is a usual practice that the breeders go for, or for radiations, where we have large mutations to really build in that genetic diversity that would be required to enhance the genetic gains in the crops that we need for food security. It is time consuming though, and there are expensive screens that need to be looked at, whether they are forward or the reverse screens we are talking. Then comes transgenesis. Dr. Mishra talked about genetic engineering. This is what is transgenesis all about, bringing the traits from other sources where they exist to the plant sources where they do not exist. However, it involves a lot of regulatory complexity. Countries have been struggling around having robust regulatory systems in place. There are issues around the safety, and it has been a never-ending saga, especially when it comes to the regulations in the regions that we operate in. And of course, the high cost, because all these regulatory data would eventually result in the cost of the technology. That is not something that public sector organization can actually afford. Of course, private sector has been investing in technologies like these. And now we have the newest in the kitty, the genome editing that everyone is talking about. What is it? We have the genome, so what? The genomes have been sequenced, so what? We have terabillion or whatever megabytes, I don't even know how to really, but then what? What are we using it for? Do we really have a direct applications other than just identifying the SNPs or the haplotypes? We still have to go a long route of bringing that knowledge and translating it into our breeding pipelines. But then we have something in our hand now, gene editing, a lot of new technologies, ZFNs, TELENs, or CRISPR are now in place that can help us getting into a precision agriculture, precise and predictable variations that can be made in the elite variety itself. So you don't have to really go back and cycles of back crossing to bring in the trait, but you can just pick your elite variety, the popular variety, and if there are the right tools and technologies in place, the genome editing can be done. Let's talk about the mutation breeding first. I will not go into detail of this very busy slide, but that is something that we have been doing. The breeders, the farmers, not farmers really, but some progressive farmers actually. I come from Himachal, and there are some progressive orchardists who actually do EMS for stone fruits, so they know how to really bring in that type of a variation. So yes, some farmers, but then a lot of breeders actually rely on mutation breeding. And if there is a trait that they identify in their screens which is tolerant to a particular herbicide, that just goes into their breeding pipelines. And here we have a new variety that has a herbicide tolerance. But then the fact is that mutation also causes a lot of substitution, deletions, insertions, inversions, and tandem duplications. And how many of us actually think about that when we have these varieties on our plate and the food that we eat for? Whether or not it has come from a mutation breeding, whether or not it has come from an EMS mutagenesis vis-a-vis a gamma radiation mutagenesis. We really don't ask those questions because we have a history of safe use on conventional agriculture, the conventional breeding. So please keep that thing in mind before we move further on this debate today. The trends on GE crops, genetically engineered crops, also known as GMOs, there has been, for last 2.5, 3 decades, there have been a lot of work being done by private sector in this direction. A lot of crops, whether that is something like a fiber crop like cotton or soybean or canola or corn and also rice in some cases. There have been a lot of genetic modification that has been done to bring some of the traits that have been bred for mostly by private sector. Not many examples from public sector research organization have been there. But this has immensely contributed to the economy. For our closer home in our country itself, BT cotton has changed the way the cotton used to be in this country. Yes, but when it comes to food crops, each one of us is, we want to be really sure what we eat has to be safe. So that food safety is something that has been extremely debated in context of GM crops, and so the progress has not been as fast as for the other non-food crops. Some of the global examples from public sector research I thought I will put here. What are the traits that have been dealt with in crops like rice, pulses, groundnut, cowpea, cassava, maize, potato, mustard, and eggplant. I might have missed quite a few of examples, but the message I wanted to give was that there are a lot of traits that have been looked at. Something as simple as insect and pest resistance, which are considered as input traits, to also as output traits like food safety. Aflatoxin, for example, where there is no variability available to breed for a crop that is safe for, that can be, safe for aflatoxin. Then what you need is a technology to really bring about that safety that we all are looking for. The most advanced technology at the moment is gene editing. I will not go into details of these examples, but you will see that a lot of work is already going on in different labs across geographies on all the mainstream crops, which are the 3 big grains, rice, wheat, and corn, to also something as smaller crop for countries like India or for the West, like sorghum and pearl millets, whether these are traits for disease resistant, insect resistance, virus resistance, or some nutritional traits or biofortification. So I will not go into what gene editing is. I'm sure the audience is aware how gene editing is done, what are the different technologies. But the fact is that gene editing gives us an opportunity to go for a precision breeding with a process that is very close to what we have been doing and what we have been seeing in nature, with a greater efficiency though. So it is a method that allows faster and much precise plant breeding. The methods used without a transfer of foreign genes, so there are methods that do not actually qualify for recombinant DNA. In some cases, there is a direct delivery of RNA by spray-induced gene silencing. There are technologies that are coming out where you do not actually have a recombinant DNA integrated, and you can still edit that. For base editing, for example, you just tweak one base pair here and there, and a new SNP is created that gives you the desired output. The resulting plants are identical to those derived from the conventional breeding or could occur naturally, and it does provide access to products that increase productivity while preserving the environmental sustainability that we have been looking for quite a while. So these are few examples, again not too many details, but there are a lot of work that is happening in institutions across the world on developing gene editing products for different crops. However, as we all know, the gene editing products do not come under one classification, like what happened with genetically engineered crops, right? So this is around, they have been categorized into the extent of editing that has happened, whether it is just a one base pair tweak, as I just said, the base editing, or there are few base pairs based on something which is a deliberate targeted transfer of an oligonucleotide or a synthetic ODN, but from the same species. species. So if you have a wild chickpea and you identify a haplotype, whether the same change can we bring in a high-yielding variety of today, and can we bring about that change that we have been looking for. However, the STN3 is still something where you would probably need much larger fragments to get into the system, the genome, which could possibly be treated as GMOs, because then the whole thing changes, and it would involve a much deeper understanding of what exactly has happened with this new change. So some of the policy consideration that we have been talking about, and since we are in the middle of a consultation on a draft policy document in India, there are certain applications that result in varieties that cannot be uniquely identified from conventionally developed varieties. Certain approaches utilize the recombinant DNA that is subsequently removed through breeding, and certain approaches actually do not use any rDNA. There are several countries that have decided to treat gene editing products as conventional varieties. For example, US and Australia, New Zealand and Argentina, and they have, based on case to case obviously, and thus the seed and grain will be handled as conventional means. The blend of companies and institutions utilizing gene editing in breeding programs is much more broad than those who are involved in GM. So the technology is much more open. There is an open innovation. There are tools and technologies that are being shared both by public and private sector. So it's calls for a much deeper understanding of what is actually happening around these gene editing technologies. However, we have to look at this with caution, since the first applications of gene editing in a country can set the precedence for what we are going to see in future. So we have to make a very, very informed decision on how we really want to deal with gene editing in agriculture applications or say health applications, because the two could be very different. So the regulation that you would need in, for example, an agriculture sector will be extremely different from what they could be in the health or in a medicine application. So those are a few things that need to be looked at. Some of the questions that I want to end my presentation with is it appropriate to make arbitrary and unjustifiable distinctions between gene-edited products and the mutation breeding products? And as policy decisions are made in this country, are we adequately considering the widening array of new approaches? So it's not, rather than putting everything in one kitty, do we really look at the technology, tools, and also the applications before we make any decisions or any regulations? So thank you so much.
Thank you, Dr. Bhatnagar Mathur, for your primer on the genome modification technologies, introducing the concept of food safety, and also bringing up the whole point of policy considerations on gene editing products. If I could invite our next expert panelist, Dr. Amitabh Mohanty. Dr. Mohanty is the Program Director at the National Genomics and Genotyping Facility, the National Institute of Plant Genome Research, which we know as NIPGR in New Delhi, and he's been there since August 2019. Prior to this, Dr. Mohanty has held several leadership roles in DuPont Pioneer at the DuPont Knowledge Center, including that of site leader for the Agri-Biotech Group. While at DuPont, the key focus area of his group was novel gene discovery for various traits, including developing competencies such as CRISPR-Cas-enabled advanced breeding. His group employed both native traits, advanced breeding, as well as transgenic approaches for crop improvement. He built a state-of-the-art and best-in-class facility for cutting-edge research and development for the organization. While preparing his bio, I was really pleased to discover that after he completed his PhD, Dr. Mohanty trained in high-throughput rice genomics at the Rice Genome Research Program in Japan, and when he returned to India, he helped to jumpstart the Indian Rice Genome Sequencing Program, which was part of the International Rice Genome Sequencing Program. Dr. Mohanty is passionate about the access of latest technologies to farmers and improvement of farm productivity. Dr. Mohanty, we are very pleased that you accepted our invitation to be part of this panel, and we look forward to hearing your thoughts on the subject.
In the interest of time, and I would like to listen to Dr. Imran Siddiqi as well, I am putting a 5-minute timer for myself. so that we can try to do a justice all across. Okay. So before I start my part of the discussion, I would like to ask start by asking a question. So what do you guys know? What is the percentage of children under 1 year that is vaccinated for some disease in the world? Is it 20%? Is it 30%? Is it 50%? Is it 80%? Any guesses? Wonderful. You don't need to read this book. So this is a wonderful book by Hans Rosling. He talks about factfulness, and he has done this survey. He has asked 12 questions, and wherever he has asked, the best percentage of answer across 12 is 30%, which he says is worse than what chimps can do by chance, right? So I'm really fascinated to be in CCMB and write this. That's the right question. So most of the places the answer gets wrong. Why? Because our worldview is like 30 or 40 years back. World has moved along, so we have made tremendous progress. Unfortunately, we don't see slow progress as progress, right? We have improved livelihood of millions of people. About 700 million people have been uplifted from poverty, right? We don't talk about those. Why I talked about that vaccination one? Because yesterday I was reading Times of India, and there was a news that Telangana is 35th state in the country in terms of vaccination. Right. Now, I'm not getting into a debate that this is right, wrong, but in general, negative news catches our attention. Similarly, when people hype about negativity, so I'm pretty sure that all of you know about the Chinese experiment of genetic editing, like gene editing in human embryos, right? Please, by show of your hand, tell me how many of you have read about the paper. Like there was a bunch of 3 papers that came about domestication of wild tomato. Have you read those papers? I see one head nod. Okay, so that is a challenge for us, right? We tend to get into the negative mindset more often than the positive. So what has happened is that in this particular example, there is a wild tomato which is known as Solanum pimpernifolium. See, all these commercial cultivar that we use today, they have gone through few thousand years of domestication. That's how they have come to market, right? So that means they are photoperiodic wise right place, their fruit ripening is the right place. A lot of different genes has to be in the right place, right time, right tuning in order to make those happen. So these 3 labs, they have done editing of about 5 different genes and was able to almost domesticate a wild variety of tomato to a cultivated variety. I said, please remember, I am a scientist. Hyping is not my forte. I said almost. This has happened in 4 years' time. This is the power of genome editing. It was absolutely impossible, right? And I have been fortunate that I started working on genome editing when a lot of people have not heard about the term, 2013. In fact, Dr. S.R. Rao, who was senior advisor in DBT, we organized a meeting, first meeting in India in 2014 in Jaipur, right? And 2017 Science Congress in Lords about Tirupati was the first time when the guideline was discussed. Both of us were there in that meeting, okay? I'm really excited to see at least, and I thank DBT and especially RCGM chair, Dr. Rakesh Mishra, that at least some guidelines have come up, right? That's a terrific accomplishment. Now we have to get into the merit, what needs to be done. Let's be debate, discuss. Like, I'm not saying that you have to follow one view or the other. I will give my personal views, and with an idea that whatever my view is, it is nothing to do with my previous or my current or my future organization. So what is the difference between genome editing and genetic engineering, right? So to me, as a scientist, a practicing scientist, it is very, very simple. We are, in the case of genome editing, we are trying to edit the native gene, right? Like I have a rice plant, I am trying to make few base pair changes. It could be few base pair, 2 base pair, or I am to delete a gene, right? I am trying to do that editing in that genome itself. I am not trying to create any new combination anywhere. So how is that different than what we have got through conventional mutagenesis? The tools that we are using to edit, and this is the beautiful thing for plant sciences, you can breed it out. Suppose you have something like, of course you have to have the CRISPR-Cas, and Cas9 enzyme has to be there, guide RNA has to be there. You can breed those out. You can only select for your mutation. which is not possible in many other places, right? So first, my request would be for the regulators is that we should separate the guidelines for plant, human, and others separately. That's a must. The other point I want to highlight that I have been spending a lot of my time in last 4 or 5 years trying to popularize the technology, because this is extremely powerful. And one of the trend that probably has not missed our attention is that our neighboring country has jumped into it full force. They have acquired a company for $43 billion because they want to have access to technology. Very recently, they have edited 30 different varieties and made them fragrant. This is what genome editing can do in a couple of years' time. So if we do not do today, we will be left behind. And I'll start my part of the discussion, and let the time for Dr. Imran Siddiqi by stating is that one of the best thing about this particular technology is that cost effectiveness. There has been scientific studies which says in order to deregulate one transgenic event globally, it takes $130 million, or in roughly about 900 crore. Whereas through genome editing, you can do the same between $12 to $15 million, which is less than 70-80 crore. Right? So all the thing that we hear about multinational getting stranglehold on this one is not true. This, in fact, if our guidelines are progressive, science-based, right way, it can allow our smaller institutions, startups to do fantastic work. And I hope and pray and wish that we are progressive and we follow the guideline, or we formulate a guideline which is going to help our young generation for taking care of our food and nutritional security. Thank you so much.
Thank you, Dr. Mohanty, for putting a positive spin on genome editing. Far too many times we focus on the negatives associated with a new technology, but now it's time to see how we can use this extremely powerful technology to make breakthroughs in whatever domain we wish to. Our fourth panelist today, Dr. Imran Siddiqi, needs no introduction to this audience. He's a J.C. Bose Fellow and has been at the CCMB since 1992. Research in his lab is mainly focused on understanding meiosis and germ cell formation in plants. His work is aimed at understanding the control of meiosis and meiotic chromosomes organization in plants and applying the information obtained from molecular genetic analysis of meiosis and gametogenesis towards developing new methods in plant breeding. His group has made breakthrough contributions to the basic understanding of clonal seed formation in plants. Their work on apomixis, which is also called asexual reproduction, has the potential to revolutionize agriculture and allow farmers to be more self-sufficient and clone their own high-yielding food crop seeds instead of buying them for each planting. In addition to his research, Imran has been very vocal in his views on GM crops and their introduction in India, and having served on several technical evaluation committees related to this domain, he has an acute understanding of the issues at hand. He addressed some of these issues in an article that appeared in The Hindu last month. Some of you may have read it. This was entitled "The Flawed Spin to India's Cotton Story." I suspect that in his opening remarks, he will be alluding to some of the things that he's written in this article. Thank you, Imran, for being part of this panel, and we look forward to your thoughts.
Okay, thank you, Raghu, and for the invitation to speak on this. So since everybody has way overshot their time, I'm not going to be shy about about taking more time than I was allotted. Okay. So what I'm going to do is to try and give a general perspective on these issues, all right? And one of my concerns also is that we all, including me, we all have tunnel vision in the areas that we are working on. And the question is, how does this impact societies in general? What are the real problems that society faces, farmers face? And where does, technology fit in the larger set of problems that face Indian agriculture, okay? So I've titled this as, what should we expect of GM biotechnologies in Indian agriculture? And by GM, I'm including both transgenic as well as the latest, CRISPR that you heard about, all right? So let me just start with the facts, okay, which has happened in India so far, all right? So BT cotton is the only approved GM crop being grown in India right now, okay? Now I use the word approved here because it turns out that over the last few years there's been very large degree of even unapproved, illegal GM crops being grown in India, and we won't get into that question, but that is happening, okay, actually. So let's go by what, what has come through the system as such. So BT cotton is the only approved GM crop being grown in India, and India has now, over the last couple of years, it has become the largest producer of cotton in the world, and it has gone beyond China and USA. And this is projected as, as a major achievement, and it's a very proud achievement. And it leads to sort of, there are two viewpoints, all right, which I have stated over here. Okay, so one is that BT cotton hybrid. So all the BT cotton that is being grown in India is as hybrid BT cotton, and for the proponents, it is that hybrid BT cotton has been instrumental in increasing India's cotton production to the highest worldwide, and this has been used to, this makes a strong case for actually extending GM technologies to increasing production in other crops, all right? That's what most of us, even biotechnologists would perhaps hold this view, all right? Now the second viewpoint, which is that of critics, is that BT cotton hybrids, require much higher inputs, and they do in fact require higher inputs, more fertilizer, as well as water, it turns out. That's because hybrids are all long duration hybrids, so they need water, they have a longer lifetime, so they need more water. And these higher inputs have negatively impacted livelihoods and contributed to agricultural distress, particularly among resource poor farmers, all right. So this is, we all know that agriculture is in a state of acute distress in this country, all right, and cotton farmers are among those who are the most distressed, all right. So this is, and nobody really quite knows, what to make of it, in these things. So, there is distress, it's going on, there are multiple factors, market factors, access, market fluctuations, so on, with the opening of the economy and so on, all right. And our prime response, our main response seems to be to write off loans that these distressed farmers has been taken. But we don't really, have much of a solution for this. And we haven't been able to come up with one. So let me actually show you this slide here, all right. And I became aware of this. So this is, this graph was taken from a paper which was published by Dr. Keshav Kranti, who was the director of the Central Institute for Cotton Research in Nagpur for a number of years, and he published this in a newsletter in 2017. And this shows what cotton productivity, that is yield per unit area, all right, over a period of times, over a period of 60 years pretty much, spending all the way up to the present. And what you can see is that so these are some of the top cotton producing countries in the world, and what you can see is that India's productivity, that is the yield per unit area, has been consistently far, far lower than that of these countries, all right? Both before BT, so this is around when BT came in around 2002, and BT has, productivity has gone up slightly, over here, all right, by about 30-40% or so it has increased, but it's much less than all of these other countries, all right, which I have listed over. So these include both developing countries. USA is not on this slide. USA is kind of comparable to this, it's over here, somewhere over here. But these countries include both developing countries as well as developed countries, all right. Mexico, Turkey, these are all developing countries. So it can't be explained just by hybrids, just by socio-economic conditions alone, all right. Now it turns out that the factor that is actually contributing to this is the fact that India is the only country in the world that is growing cotton as hybrids, all right. So hybrids, as you may know, are made by crossing together two parents, all right, whereas varieties are ones in which the seed from that can be essentially reused again, all right. So these are true breeding, whereas hybrids are not true breeding, all right. Now all the hybrids, India has been growing hybrids since starting in 1970. In fact, it was the first country to come up with cotton hybrids, and it gradually started shifting towards cotton hybrids. All right. Now, by the time BT came in about 2002, about 70% of the cotton being grown in India was hybrids. All right. All right. Now, BT cotton was introduced into India only in the context of hybrids, and since it was proprietary technology, that essentially took over, and essentially all the public sector cotton seed production stopped. Okay, it all became BT, and BT cotton was pushed very hard, all right, at that point. But it was all as hybrids. Okay. Now, it turns out that all of these other countries have been growing cotton not as hybrids but as high density compact varieties. All right. So these are ones in which the number of bolls per plant is somewhere around 5 to 20 bolls per plant, whereas in hybrids, which are sort of bigger, bushier, they need more water, they are long duration, these produce about 40 to 100 bolls per plant. But these are planted out, these compact varieties are planted out at about 10 times the density in the field that these, that hybrids are being planted out. All right? So this lower boll production per plant is more than made up for by the fact that you're getting much higher density on the field. All right? So the yields that they are able to get is around 1,000 to 2,000 kilograms per hectare, whereas the hybrids are giving you, and what India has achieved is around 500 kilograms per hectare. Right? So that's half to a quarter of this thing. And we have been consistently following this thing for 40 years, all right, both before BT, and BT came in and was essentially dovetailed into this hybrid seed model. All right. Now, obviously, the reason why BT came in that context is because hybrids allow the company to retain control of the seed as well as pricing and so on, because the farmer has to buy the seed at each generation, at each time, whereas varieties can be reused. So in these other countries, even when BT came in, what you see, so let me show you this thing. So this is another interesting point, is that it's possible to actually achieve this high yield even without BT, and this is shown over here in the case of Brazil. All right, so Brazil around 1996 or so, no, not 1996. Sorry, 1996. It shifted over to this high density type strategy, and what you can see is that there's a dramatic increase in the productivity. All right, so it went up threefold or so. Okay. To hit around 1,000 or so within the space of about 4 years. Okay, and this is non-GM. This is, at this stage, it was non-BT. It was not BT. These were just high density planting variety. Later on, BT was added to this thing around 2012 or so. All right, or before that, maybe 2010. But in any case, this yield, this large increase in yield that happened here was not because of BT. It happened because they shifted over to this compact type variety. All right. We, on the other hand, have been continuing with this thing for 40 years. All right, and it's I still don't know why this is the case. All right, but this is what we've been doing. And it doesn't make any sense at all, but this is what we've been doing. All right. Now, you can see that this is just as a comparison of some of the properties of hybrids versus BT cotton varieties. I'll just draw your attention to the fact that these cotton hybrids uses much more fertilizer, about twice the amount of fertilizer that the compact varieties do, and they also have longer water requirement. More water requirement and later in the season, because these are all long duration varieties. All right, they flower later, so they need water later in the season. So both these features, more inputs and more water, pose greater risk to farmers. All right, particularly late in the season. All right, so if it's grown in rain-fed areas, and about 65% of cotton in India has been grown in rain-fed areas, low-resource farmers, all right, those farmers are put at greater risk by using these cotton hybrids. So if you have, enough resources, enough water, you can probably get by with these BT hybrids, all right. But if you don't, then you don't even recover your costs. So your costs are greater, but you don't recover your costs because you don't, at this critical period when the cotton boll is forming and water requirement is maximum, if there is no water, then you don't get that, all right, in these things. So it's very likely, of course, the data on this is fragmentary, but it's hard to, it's just anecdotal data which exists, but anecdotal data there is anecdotal data that, greater distress is seen among cotton growing farmers in these rain-fed areas, all right? But large-scale macro data is not available, all right? So what we see, the picture of BT cotton in India viewed in an international context, it's largely been a profit-driven application of BT technology in highly suboptimal contexts as hybrids, okay? And this was kind of layered onto a pre-existing use of hybrid cotton in India. So you have increased input costs, somewhat greater yield, and high risk for low resource farmers in rain-fed areas, all right? There are some other additional factors also. You have pink bollworm resistance, which is actually unique to India, all right? And that's because it's a long duration, which is when pink bollworm, infects cotton. So there's increased susceptibility to disease also in these long duration hybrids. So why have we persisted for so long when all other countries have been, going with these, essentially ignoring international data, is a mystery remains to be a mystery to me, all right? So one important take home from this is to recognize is that the outcome of a technology is determined by the context of its application. It's not determined just by the technology itself, right? If it's deployed in a suboptimal context, you don't get, good results from it, all right? So now this has been, it's remarkable that this has somehow, so all the increase in cotton that has happened in the case of India is obviously because of much larger increase in area, all right? It's not because you've gotten better at growing cotton, all right? So what is the context of let's see, if you use the word context in a larger sense, including the socio-economic and the larger, features of India, what you have is you have very diverse agro ecosystems in India, all right? Very, very diverse. There, the estimates that you have something like 65 different agro ecosystems in India, all right? You also have, very small land holdings in India, okay, these things. Now, what is the place of agriculture in Indian society, all right? What you can see, what is clear, everybody would know this, is that, you have a very high percentage of people whose livelihood depends upon agriculture, about 60% or so. But the contribution of agriculture to the GDP, if you decide, if you look at it in those terms, is proportionately much lower. It's only about 25% to the total GDP. So these are, low income generating processes, but it's subsistence farming. So you have a high percentage of these are actually subsistence farmers who are developing, who are dependent on agriculture, right? So the issue then becomes really sustainability itself. The question of increasing productivity, these kinds of arguments you have to be cautious about, because in the name of increasing productivity, doubling, tripling income, whatever, all right, if you come up with varieties or, strains that are high yielding but require higher input, you actually make things worse, particularly for resource poor farmers. If they're not able to put in those resources and they don't have access to those resources, or those high yielding, in most cases high yielding productivity increasing varieties do require more input, all right? We also have very poor infrastructure. India has not invested well in developing, good storage facilities or even transport, all of these things. So, you have very very old and inefficient systems of market yards where the farmer has to sort of somehow get his produce there, and there's room for manipulation of prices. Following the Green Revolution, there's been a complete erosion of extension services. So even advice to farmers has deteriorated badly, all right? Much of it is just informal, often taken over by private, the suppliers of seeds themselves. So it's not something that is operating necessarily in the interest of the farmers. It can be operating again through profit-driven, factors, all right? And in the case of cotton, what you also have is, high risk. There's very high credit costs. Loans come at very high interest rates. We don't have systems for agricultural loans. Farmers are paying often interest rates of about 35, 36, between 30 and 40%, right? If they don't have collateral, poor farmers. These are very, very difficult conditions under which they are being asked to produce, right? In these things. Okay. So what are some of the lessons from this thing? So one is that it's very important before releasing GM crops or anything of a GM product. I don't mean just the technology here, but you need to be able to assess the product. You need to be able to assess the suitability of the GM product across a cross-section of farmers as part of the regulatory process, all right? That means socio-economic considerations. Now it so happens, I'll come to this in a minute, that the international agreements that India has signed do provide for inclusion of socio-economic considerations in GM regulation, but these considerations are completely missing at present from the Indian regulatory system, all right? It's just not there. It doesn't exist. It's astonishing completely. You've also had, replacement of public sector seed production, the state seed corporations by private seed companies, particularly for hybrid seeds, because these hybrid seeds, hybrid BT in particular, was a proprietary technology. It was not accessible to the public sector seed production at that time. Weakening of farmer advisory and extension services. The other factor that we need to keep in mind is, and this is a general factor, is that the high cost and time of development as well as regulatory clearance for GM products. I mean, somebody mentioned the figures of 13 million or $130 million. Those are huge figures, all right? They do not address the diversity, the number of products that are out there. So how many products are you going to actually develop at that cost, all right? Now, once you have obtained regulatory clearance for something like this, obviously the company or whoever is doing it has to recover that cost, okay? And that means that they will push that product very hard, all right? And so only developers with deep pockets, and the Indian public research system does not have such deep pockets, all right? Let there be no mistake about this, all right? All right. What it favors is the private, is private companies on these things. They are the only ones who have these kinds of resources, all right? All right. So this favors developers with very deep pockets, and leading to overuse and monoculture-based approaches, and that additionally poses risk. So particularly in the case of disease resistance, BT and so on, that leads to breakdown of resistance. And that has in fact happened. So both, Bollgard 1, Bollgard 2, both their properties have become undermined because resistance has emerged, all right? Again, partly because of use in suboptimal conditions and overuse and misuse also. It has not been used properly, all right? So high cost of development favors overuse and monoculture, leading to breakdown and unsustainability of the product and technology, all right? So it works very well for 4-5 years, all right? After that, you have to go back to using insecticides more and more. So that is in fact what has happened, and it has been the international experience in the case of BT that for the first 5 years it works, then as resistance starts to arise, you have to use chemical pesticides more and more, and the usage of as the resistance increases, you have to start using more of those pesticides, and after some time you're back to where you were even without BT, all right, over a period of 10 years, all right. So, of course, this is, so one thing so what does one learn, what is the perspective, what is the take-home message from this thing, all right? So one is that, excessive reliance on purely technological solutions such as GM to solve agricultural problems, ignoring wider issues can distort policy, all right, in this thing. It's very important to try and get a grasp of this thing, and this is true for us also as biotechnologists, as molecular biologists. We are so interested, we are so taken up with the technology itself. We think that this technology is somehow going to solve these problems. But technology is just a method, all right. The question is, what are you going to do with that method, all right? How is it being applied? In what way are you applying this thing, all right? So in India, basically reducing risk to farmers is probably at least as important as increasing yield, all right, in this thing. So you need to be lowering input costs to be able to make it economical for farmers to at least be able to grow. And there are many other things, there's infrastructure, increasing farmer incomes, making credit available at lower cost. These are major problems, and they're nothing new. They have been pointed out by the Swaminathan Commission report in 2006, but essentially nothing has been done about this thing. So it's important to recognize that GM does not function in a policy vacuum, all right, in this thing. If you have a policy vacuum, the results from GM technologies are not going to be favorable, okay. The other very important thing which I have written over here in red is that the scope of GM regulation in India has to be expanded to include socio-economic considerations of the product, okay. This is just not there, and although there is the treaties that India has signed, the Cartagena Protocol explicitly includes mention of socio-economic consideration, but it's not being applied in India, all right. Major problems such as climate change and reduced groundwater availability, and all these are these are other factors. And these pose complex challenges, all right. So it's not clear that a single product or a single gene point approach is going to address that. You have to have a wide set of practices and context, to address these things. We need to pay greater attention, as traditionally we have been in the past, towards long-term sustainability as opposed to short-term gains, all right, in these things. In the past, many traditional varieties, locally adapted varieties were grown, and their yields may not have been as much, but they were steadier, okay, and they worked in those areas. Many of these have now been sort of rejected, basically because people have been using high input agriculture, and people are favoring increased yields, but sustainability and all has suffered over time, right? So these are things that one needs to consider. I mean, I'm not saying that you just go back completely to everything else, to what it was before, but you need to consider these factors, all right? I didn't talk about herbicide tolerance. There's no time for this thing, but we basically, you need, even for pest resistance, it's better to have combinatorial practices, all right, rather than using single point solutions like BT or herbicide tolerance. And even the US National Academy of Sciences, at least in regard to herbicide tolerance, they have actually explicitly recognized this thing, that a diversity of methods is actually more important for sustainability than single point based solutions for this. Yeah, I'm okay. So this is the Cartagena Protocol, which talks about socio-economic considerations, all right. And ending over here, let's see. So get back to the question: what should we expect of GM biotechnologies in Indian agriculture, right? It's very important to recognize that GM and CRISPR are tools for research rather than technologies for products, okay, application. We have to keep that in mind, okay. The purpose, the goal is not to be able to come up with a GM product or a CRISPR-based product, but to understand, use it to get information, about the plant or whatever, so that then you can use it in whatever way you want to use it, right? And that, in fact, is how it has been used. But somehow this idea that you have to come up with a transgenic or a CRISPR-based product as such seems to have also been distorting the research environment itself, right, in these things. So if the product is so, if the technology is so good, what you need to really ask is, are there problems in agriculture which can only be addressed by GM or CRISPR-based thing, right? That needs to be a test that you need to ask. If the same problem can be solved by simpler methods or alternative methods, all right, it makes sense to actually use those methods rather than spend millions and millions of dollars trying to, trying to come up with a CRISPR-based method for this, all right, in this thing. So I'll stop there.
Thank you.
Thank you.
Thank you, Imran, for your views. That's how we should learn from history on how to go forward. May I have the panelists back on the stage? We have maybe about 10 minutes for discussions or debate. In the audience, I have some questions here. If there are no questions from the audience, I can start here. Are there any burning questions from the audience? Any? Yes, okay. So there's Sachin. Can someone give Sachin a mic? Please identify yourself and tell us who your question is addressed to, or if you would like the whole panel to respond.
My question to Imran, that as you showed in the
Mike.
Hello. My question to Imran, that you showed in the graph that Brazil picked in 1996 that almost production of the Brazil and India more or less same during the course of the time, but Brazil picked up and jumped many-fold, but India, despite of the opting the BT, is unable to come up in that range. Why India failed? Is because of the policy making? Is because of the...
No, it's because they continued with...
Hello?
Yeah.
Okay. We essentially continued with hybrids. We didn't shift to these high-density planting systems. So that steep increase that happened is when Brazil actually shifted to high-density compact type varieties, whereas India didn't do that.
No one knows.
Yeah. Well, that's. I mean, I asked that question also, and see, I mean, you can speculate, all right, that India was, we were in love with hybrids because we had invented them, or else, there was influence from whoever. Now that's a speculation, I mean, I don't want to say, I don't want to get, but the fact is that, we didn't do it.
Okay, thank you, Sachin. Any other questions? Okay, so there's a related question that was given to me. It says, what has a greater impact on influencing policies on GMOs, science or politics? So maybe the panel can comment on this, because it seems that many of these decisions are taken by politicians or people who don't know much about the technology. So I'd like the panel's views on whether they believe this is true or not, and what is the way to move forward.
I can give the short answer, then others can give more technical. I think this is, I have the mic. So this is the misuse of or lack of rigorous science by technology or companies or something, because it's not failure of science, it's not using science properly. That gives a scope for politicians and other influencers to influence the process.
Anyone else on the panel would like to take this on? Okay, sensitive issue.
Okay, I mean, I can.
Please.
Because I've seen.
Use the mic.
See, the thing is that, if you look at it, in India, the regulatory system has been, very limited, all right? Now there are actually, what is the regulatory system? Internationally, there are treaties that India has signed, which provide the broad principles of what a regulatory system needs to address, for transgenic crops and so on. All right, so those are there. Now it's up to each country to develop it. And in India, I think what has probably happened is that, there was an expectation that, and that was true, even before, soon after plant transformation and, transgenic technology became available, that because of the potential that it offered, the ability to transfer genes, a whole wide range of, products could be developed which would address the needs of agriculture, including Indian agriculture. Right? So there was almost an assumption, and many of the companies that were first off the block, like Monsanto and so on, everybody genuinely believed that that this is going to be the next great thing, and they were able to drive, and some of their products have been commercially extremely successful. BT and herbicide tolerance, that is where they have made their money on these things. Now in India also we tend to take that on as received wisdom, and we didn't really do our own assessment in detail as to what is it that is required. Now BT works, all right, for the first few years. There's no question that it does, that BT works, all right. The question is that we kept on with hybrids, and nobody in the regulatory system actually asked what form is it actually going to be deployed in, all right. So I don't think that it's politics. In fact, I would say that Jairam Ramesh, at the time that BT brinjal was was approved, he had his reservations, and he actually sent the regulatory documents out widely, including to the National Academy of Sciences, USA, who appointed scientists to look into those products, and they provided critical inputs to this, all right, very, very detailed analysis, all right, and it was on the basis of those inputs that Jairam Ramesh actually put a moratorium on this thing, all right. So we have been kind of largely drifting along, is what I would say. It's not politics, all right, it's our own, the scientific and administrative also. I mean, we just have not invested in regulation. We've assumed. And even policy-wise, I would say that, even within the departments itself of DBT and all of these things, we have become so identified with, with the process of coming up with transgenic products that it's almost an assumption, the purpose of research is to be able to translate it and to, make a transgene and come up with a transgene, all right? And, it's interesting to ask this because, for many things, there are many papers out there where people took genes, say a drought resistance gene, something goes up, under drought resistance, you make a transgenic, you find some sort of an incremental response, increase, and everybody gets excited and you start saying that, you're going to get into more and more trials of these things. Now, that turned out to be the case for many, many genes, all right? And what became clear over a period of time was that, drought resistance is not a single gene, it's a network response, all right? So you can take any element of this network, and once you increase its expression, you start triggering that network, and, the plant starts to behave as though it is under stress, somewhat under stress, and that confers a certain level of tolerance to this thing. But that does not translate necessarily into field level tolerance, all right? So as researchers, I think that the primary responsibility lies with both researchers as well as this, the research system as a whole. It's not politics. I don't think it's politics.
If I may give, I'll give my take on it. First of all, I don't have any knowledge about cotton, okay, and especially BT cotton. I'll talk about rice. Right, I see one of the reason our productivity has not improved or increased is because we have been using the same genetics for I don't know how many years. So I saw that graph And what occurred to me was that there was a doubling of productivity from 250 almost to 500, and then it stayed stagnant. So you can trace it back to, like, we have only released one event, right? And that is being recycled. If you go to Western countries, and again I'll talk about why I'm talking about rice. Let's look at the rice. Why hybrid rice in India is only 6% of the cultivated area. 94% of the area is still under hybrid, in the cultivated rice. Because our public system has done terrific job of improving yield. Right? They have been breeding newer varieties, they have been releasing. So every now and then, Pooja Bhatnagar, I did my PhD on Pooja Bhatnagar 1. I was the first one to make transgenic rice in India. Okay, long time back, 94, 95. Since then, there has been a lot of different improved varieties have come, right? So our public system has been doing a terrific job. I don't think that has happened in cotton, like, with my limited knowledge. Probably that is one of the area that, we need to strengthen our system. And I agree with Dr. Imran is that the public system, need to be definitely strengthened. No, there is no doubt about it. Probably some of the problem that we are having today, we can trace back to, our lack of proper R&D, improving genetics. If you don't improve our genetics, corn productivity has gone up 1% every year for last 40, 50 years. Every year 1%. So you may not see year to year, but over a period of 10 years, you will see huge improvement. So that is something that I would request everyone to pay attention to.
Thank you. So Dr. Bhatnagar would like to chip in.
Yeah, I would partly agree with my fellow panelists, but I would like to just offer a little different perspective to it, right? So the question was whether it is politics or it is science. If I have to answer this question, I'll say probably it is both. Because, we are looking at two different things. We are looking at regulations for a technology, but for some reason, and which is strange, I really sometimes don't understand, we start looking at other things. Socio-economics and regulations are different things. So, for example, for medicine, FDA approves a molecule or a drug discovery. At that point in time, the sole purpose is to look at the safety of the molecule. Whether the molecule or the cancer drug that comes out of it is 5 rupees or 500,000 is something that is probably not looked by the regulators. So I think there has to be, yes, both are important, but it's the mishmash it causes, because we look at regulation, which is a science-based regulation that needs to be done. At the same time, then we start looking at the socio-economics, which is important. I'm not saying we have to do without socio-economics, but I think this is something that always creates a lot of confusion. So I would assume, as a scientist and fellow citizen of this country, it is a responsibility to look at both the aspects, but I don't think regulatory agencies should be looking at the socio-economics. There has to be an add-on system in place that also looks at, for example, the beautiful example Dr. Siddiqi gave on why we are still moving ahead with hybrids. That's a classical question. But do we really need regulators to look at that thing? That is something that we have to really, have an opinion on, and probably more deliberations.
Okay. Thank you for that. We have time for perhaps one more question before we close this. I wish we had longer. We could go much longer. Yes. Somebody give her a mic.
Hi, I'm Deepika. How about applications of GM crops such as edible vaccines or other plant-derived products, and how about the applications? Can you comment on that? Not just the aspect of productivity or growing them as food crops, but the other applications of it.
So the question is, please comment on other applications of GM crops where additional traits in addition to productivity are included, for example, to make edible vaccines and things like that.
I will not talk about edible vaccine, but being part of the task force member, we do see a lot of different projects, and some of them are like improving provitamin A in banana, right? So things like that has been attempted, and they are pretty successful in the lab or greenhouse stage, right? They are yet to see, because we have started quite late, so only last 2 years I see CRISPR-Cas genome editing work happening in the country. So we have started late, but there have been some researchers who are coming up with solutions like that, So it will take few more years to go, but there have been thoughts along those lines. I am not aware of if anybody is trying vaccines.
People are trying vaccines similarly, but I'm not sure whether it has reached any level where somebody is trying to trial or something. Yeah.
Dr. Shivaji has a question.
Like production of things, whether you are producing a bacteria or using in plant. If you're asking whether they are being produced, so of course it's possible, but whether rules or regulations, policies should be of different kind, I don't think so, because after all, it's the same thing, same market, same target.
Dr. Shivaji?
Let me start by saying that I really enjoyed what the presentations were about. But the question that comes to one's mind, even as a person who's not a scientist but a literate person is, do we have a need for some of the GM crops which we are trying to develop? The last slide of Imran says GM technology and genetic engineering is very good for research. 100% I agree with that comment of his. But when you try to push that to something like, just say, a brinjal, a banana, a papaya, which comes in the daily press, what is the need for our country to get into those technologies? So the question actually is that those people who are into regulation of this, are they trying to address issues as to what crops are we actually going to target, and not everything which comes our way? That's the question.
So maybe I'll take the first cut. So you are absolutely right. And I agree with my fellow panelist as well. This is just a technology, or you can call it as a tool, right? So anything that is for me it is very, very clear. Anything that is possible by traditional breeding, I would like to do it. Okay, I am not going to go CRISPR-Cas way. Anything that is possible by any other means, easily. Why I need to go and spend my money on? I am talking about only those applications which are not possible otherwise, or which are otherwise time consuming. Then you use technology. And a small example, today we import edible oil worth 70,000 crore rupees, and please remember, this has reduced. It is reduced by 10,000 crores over last few years, right? If the traditional resources that we had, and just to put in context, we can do a direct benefit transfer of 7,000 rupees to our 10 crore farmers with that money, what we are paying to Malaysia, and now is going to be Indonesia, right? So this is the impact. Now, if our traditional technologies, those are at our hand, if it was so efficient, if it was able to address our problem, then we will not have this productivity problem today, right? We are importing edible oil, we are importing palm oil, right? Huge amount, which is unhealthy, and we are consuming it because we are not able to produce, because the means and resources that we have, whether it is genetics or technology, breeding, we have not been able to solve the problem, which may not be the case in other crops. I am just talking about mustard. So I completely agree with you. If there is another resource available, why you want to go try something new, unless it gives a unique point. So absolutely, you are 100% correct in that one. I agree with you.
Okay, maybe one last question. Puran has a question. Yeah. Okay.
So, very good discussion. So my question is, so since we are talking about GM crops and still India doesn't have, but so how much GM crops would risk our traditional varieties or traditional seeds, which I read sometime in newspaper that there are many traditional varieties, the seeds are not even available. So should there be under organization or under initiative to preserve the traditional varieties and their seeds?
So if the audience didn't hear the question is the entry of GM crops or the application of GM crops in India going to affect our traditional plant varieties, and is there a move to preserve those traditional plant varieties as seeds or whatever other resource?
Okay, I can have a first one. So I think it's true that there has been a general reduction in the usage of traditional varieties in rice and so on. If you go to even West Bengal or many of these places, but that has not been driven, I would say, necessarily by GM. It's just because of favoring, high yielding and high input fertilizer based. You see, many of these varieties were grown with low fertilizer applications and limited yield also. They were often grown for subsistence. The breeding that happened was also for improved yield, but which required fertilizer-based application also. So these, once they came in, then the traditional variety reduction, usage decreased. Now it's true that because if you just take cotton as an example, okay, in this thing now, at independence time, and this is remarkable that India is a center of origin for cotton, all right, this desi cotton, which is a distinct species, and now that produces extremely high quality fiber, but it is short staple, so it is not used for the industrial mills and so on, in these things. Now that level of production, so the British actually wanted, were pushing this in India, but when the British left, less than 10% of the cotton being grown in India was in fact the American long staple cotton. Most of it was still desi cotton, right? It's when the Indian industrial mills came in, right, the large industrial mills came in, that desi cotton declined, in India. Now, still, when BT cotton came in around 2002 or so, that is when it came in, about 30% of the cotton grown in India were varieties, and about somewhere around 5 to 6%, or somewhere between 5 to 10% of that was desi cotton. All right. Now, less than about 2% of cotton is desi cotton. All right, in these things. But there is potential actually for going back to desi cotton now, because people have actually been able to cross in the long staple trait into desi cotton, right? But having gone over into this model, that we have been following, it's going to require some effort to go back. And the desi cotton actually in India is far better adapted to Indian conditions. It is more pest resistant, many of these things, all right? And it's similar in architecture to the compact type also. But policy has to be taken up, and there are problems there, and in these things. So I think fixing problems is always requires effort rather than continuing with what you've been doing, even though that's the wrong thing.
Thank you, Imran. And with that,
I just wanted to say that it's like the cars that don't have navigation system will disappear because there is a better car equipped with navigation system. Simple is if there are other good features in the car, put navigation system there also. So you enable local varieties or desi varieties with the technology so that everything else is preserved. If technology is safe and if it is cheap and easy, which is likely to be the way things are improving. So otherwise, of course, if something is more superior in terms of production or cost, that is going to dominate, it will wipe out, and that will be, I think that will be a disaster. We have already faced like a practical extinction of so many varieties. We shouldn't be doing that. The other thing I just wanted to add was that we should not be limited only on whether it's needed or not now. If you create possibilities, then you can choose, and we can be in a better position tomorrow, like what she was asking. I was in one discussion, and people were asking, if it is possible to grow vegetable without insecticide, why we are being pumped with insecticide? Why not use insect resistant? It's a very pertinent question. Audience, the consumer can ask, why if there is technology we are not given that? Do your experiment, figure it out, make your policy, but give us vegetable which we don't have to eat with insecticide, which you see are going to become problem. So it's a very complicated issue. It's not simple in the.
Thank you, Rakesh. And with the panel's permission, I shall now close this discussion. We could go longer, but then we have other things to do. So let's all thank the panel for their valuable inputs, their insightful comments, and their time, and for participating in our discussion. And we have